I don't know about all of you, but those were very inspiring opening talks. Thank you so much. But first of all, welcome to the Evergreen City. How did it get that way? Right? Everything's green here for a reason. It rains all the time. You know what they say about Seattle? It's like being married to someone truly beautiful who is sick much of the time. So I'm sure you'll get to experience her flip side over the rest of the weekend. I took a look at the weather forecast. But anyway, welcome. Really glad you're all here. So, you know, carrying on some of these incredible themes and discussions that have been presented already, I want to approach each of you from your clinical perspectives. Do you have everything you need to treat your dry eye patients in your toolkit? I think that's a no. Right? We're begging for more. So do you wish you had more point-of-care diagnostics?
I think that we definitely do not have everything we need to take care of our patients. And I think you name it right. It's not only the treatment, but also how we identify, which is where this big issue comes, right?
Yeah.
You know, it's just not dry eye, like one of your patients said. I think we need to move in that direction. We need just not dry eye, but a first and a last name. Because we know that, you know, dry eye, it's a big, and I'll mention this later, it's a big umbrella. We just put everything there, and all of you know this. So it's how we define that, and that will certainly help with what I'm dealing with and how to treat it, and to also educate patients. And James, you put it perfectly. You know, we have all these conditions that are called dry eye. Patients go to the pharmacy, they see all these drops that we tell them to take, or other providers, by the way, you're right. And of course, confusion and poor compliance and no response is the result. So we need a first name and a last name.
I love that. Or differentiate the Gen Zers into 3 different groups. Maybe that'll help. I always tell my patients it's dry is like a big circus tent. There's about 30 different animals in there, and the lights are off. Be careful where you put your hands. We need the power switch. We need to be able to turn the light on so we know which animal we're dealing with, and then name the baby or the monster, Godzilla in the crib, whatever this is. So the first name, last name, to Dr. Perez's point. Oh, I appreciate that. Thinking about diagnostics, and I know, Dr. Downie, you are doing some really exciting work about the metabolome, the secretosome, the proteome, like beyond the genome that I know that Dr. Perez is involved in as well. Like, how does that interplay into the future of dry eye, and what do you think that future looks like?
Yeah, so my personal interest is in this diagnostic space, and so I'm a clinician scientist. I lead a laboratory at the University of Melbourne focused on the ocular surface, and kind of speaking to what has already been mentioned about multifactorial umbrella term. We've kind of been looking at it from the approach that we know that there is this loss of homeostasis of the tears, as in TFOS2 definition. But there are many, many reasons that can be the case. And so one of the challenges is, you know, first determining accurately that we don't have homeostasis, then trying to work out what's causing that loss of homeostasis. So really looking at the pathobiology, how can we determine for a given patient accurately and quickly what's driving their dry eye, and then also targeting the mechanism of action of drugs to that phenotype. And so trying to piece that all together. And so some of the exciting research in kind of this omics space is that we can now use molecular information, or kind of building on that, to really understand the biological process at a patient level. So kind of a precision medicine approach where we could diagnose, prognosticate, even prescribe drugs by understanding with sophisticated techniques what's actually happening in the tear film. So there's a lot of exciting research in that area. It is challenging because of the need to standardise methods. One of the colleagues I work with, who's a protein chemist at the university, kind of jokes about what the patient ate for lunch. You know, did they have a hamburger? Because their metabolome might be slightly different to whether they had sashimi. So it's kind of a trivial kind of example, but what it speaks to is that it's very complex. And so we need large studies with really standardised methods so that we can actually extract that very powerful information that gives us the potential to deliver that precision care.
What is your prediction on how far some of this incredible work you're doing is before we have it as a point of care, so we can really help differentiate what wild animal we're dealing with the lights turned out in the circus tent?
I'm not sure on time frame, but I think we will get there. So certainly the ability to analyse, to collect tears, to analyse tears, to use the information in tears is being recognised as an increasing reality. And so we're already seeing some point of care diagnostics that do that type of analysis, but I think as technology improves, we're going to see more sophisticated approaches, really understanding the biology of the tears. Our work is looking at the behaviour of tears, so how we can actually capture biophysical properties and what that tells us about the composition and the health of the tear. So I really think that is the future. New point of care diagnostics that give us that patient level information accurately and rapidly to then direct therapy. So it's really about directed therapy.
You can tell your radiologist patient that she's going to have her wish, that people are working on it, something specific for her, right?
Absolutely, and I think it is about that translation. There's lots of exciting technology coming through, and it's fantastic what you can do with a PCR machine, etc. But these are big, sensitive, really expensive bits of kit that we can use in a university or an industry environment. But it's how do we then convert that to point of care products? Because that's where the real power is in terms of allowing the clinician to narrow down and pick the right treatment. But we are definitely seeing good strides in this area, but it is complex to do.
I was just to be the resident contrarian. You guys.
We'll have to share.
Oh, there we go.
Okay.
So just to be the resident contrarian, I come at this from a different perspective. I was an academic ophthalmologist for 15 plus years, and now I'm in an academic private practice. And just having seen, various technologies, various treatments come through, I think back to like macular degeneration. What is the one thing that unites macular degeneration, diabetic macular edema? It is permeability of the vasculature. And so really we didn't have anything to treat these patients prior to the introduction of the anti-VEGF treatment. So I guess the way that I look at it is what is the one unifying thing within dry eye, and where is that blockbuster product going to be?
I think that, I call this our nerdy approach, which that's what we are. I'm also a clinician scientist.
That's a high compliment though, right?
And then, that's our job in a way, being in academia and trying to identify this. But, the really key point is how you take it to the practice. And that's up to you too, right? It's not just developing drugs, but also developing that diagnostic test that's going to help you identify your responders or your poor responders, how you do clinical trial, and how you understand that your drug actually works and that it makes a difference. So this is a field that a lot of companies they don't want to invest into diagnostic testing. They just want the drug to get approved and move forward. And I would say that's the first step why they fail, because we don't know the first and last name. And in academia, we're not going to be able to develop this to a dumb technology that can be used by everyone. But we can develop and identify the right genes, the right cells, et cetera, that can open. It only takes one discovery to change the world. And I think that's, we're a young specialty. I mean, we're not like glaucoma or retina has been there for many years. If we think about it, we just were identified maybe in 2003 after restasis. And so, don't give up. I mean, I still think that we have opportunities to navigate these painful waters. But I think until we really understand how we diagnose, follow, and monitor, it's still really a challenge, but it's a challenge that can be accomplished.
And there's an important component to everything that we've just said, and that's like the treatment and where these point-of-care tests relate to clinical trials, right?
Exactly.
You have a heavy clinical trial site, and there's so much relevance into defining first name, last name.
That's exactly right, and I want to add something to what Dr. Perez said initially, and that is the term dry eye itself is not a diagnosis. There's much more that goes into it. And I believe that too often, because I'm in the clinic every day and I'm around optometrists and ophthalmologists, and we all are guilty of oftentimes using the same generic treatment approach to our patients because we don't, frankly, have enough chair time to really do all the testing sometimes that's needed. There's 3 patients waiting down the hall for us at that moment, and so we're guilty of kind of using the same cookie cutter approaches, which is the real reason why we need more specialized medications in the cabinet, in the sample cabinet to choose from. But yes, there are, I believe, some gaps too that exist between clinical trials and the real world setting. I believe that when patients come in for our study visits, we have absolute control really over that patient. They're following a strict protocol, a treatment regimen, but we really don't know what's happening when the patient leaves our clinic. They leave with a nice paper diary, but are they filling that out in the parking lot before they come up for their next visit? We want to know what's really happening in real time, and in a little while we'll be able to show you a incredible device that's been developed by Aura that is really going to enhance our ability to reach some of those patient-centered outcomes through subjective questionnaires, through quality of life improvement measures that can be put in place that will allow us, hopefully in time, to develop those specialized treatment, specialized medications that will give patients a faster onset of action, better tear production, all the things that we're looking for when we see patients back for follow-up visits and we hear, oh, I didn't really, fill that prescription you gave me. We hear that every day. It was too costly. I didn't feel it was effective. It didn't really improve my symptoms. And so I think, again, with what we're hearing from some of the other companies in one of the sessions to come, I believe there is great hope on the horizon for all of us.
I love it. So that, and to your point, like when you're seeing patients in the context of a normal clinic, you're right, you're super pressed for time. You have to, maybe not spend as much thinking time as you would have liked to have. I love doing clinical research because you get to slow it way down, right? And try to look at one thing at a time and see what's happening. But we need to move faster than that. We need to find the problem much faster than that. I love the idea of having better characterization of these different subtypes, these different animals in the circus tent, and then targeting specific therapies. Theoretically, this can be translated also into significant cost savings for clinical drug development, because if you've got a more sensitive and specific sign than Schirmer's, with a poor positive predictive value unless things are really bad, and you've got other things you can go off of to measure your treatment effect, you could do this with smaller number of patients, and that represents tremendous savings for study sponsors, pharmaceutical startups, seasoned startups, et cetera. So I'm looking forward to that piece. Anything else to add on that bridge? Because basically what we're talking about is from bench to bedside and back is what we're talking about, and how every little thread impacts the other part of the spider's web of this problem that we're also engaged in trying to help figure out.
The only thing I would add is that I think we need better metrics. I mean, that goes without saying, but I'm certain, myself, and I'm sure many on the panel have been involved in clinical trials with products that actually work, but they failed to meet the primary endpoint because our metrics simply weren't good enough to identify.
Yeah, great drug, but not looking at the wrong target. Yeah. No, I agree with that.
Yeah, and just speaking to that about the right population, I think what we're describing is, the opportunity to enrich a clinical trial population with those that are actually most likely to respond. Mm-hmm. And so we need better tools to first identify that subgroup, and then having that tool, like you say, we can have more precision on how we measure things, there'll be less variability, and so smaller trials. And so that will be a fast track as well to get the right novel therapies to the right patients. So I think the biomarker aspect is absolutely critical, and there's also an opportunity for diagnostics and therapeutics to kind of work hand in hand to really get the right things to patients.
Yeah, and on the other side then, when that's on the market and prescribed, we can actually target the patients, so the drug gets a much better reputation because people who are prescribed it actually are the ones who are going to benefit from it, and likewise they're the ones who are going to stay on it because it has an impact on them. So the win is all the way through if we can get this right.
Truly, I'm so glad you brought that up, because as a clinician, when a new drug gets approved, we're all excited, we're excited to try it, but what is human nature? To try it on your worst case, right? It may not necessarily be someone that they were looking for in the control within the clinical trial, and they may not perform as well and may not get the the uptake and embracing that you would have liked to have seen, that it deserves, frankly, that the drug deserves.
And also the practitioners lose confidence in it because it's not actually helping the patients that they hoped it would do. And as you say, that worst case that you really, it should make a difference in this one, or it's not. Well, do I prescribe it or don't I?
Yeah. And then asking more questions about, well, why didn't it respond? And what do I need to understand here? And do I have some tools that can help me figure out which one of these patients would benefit most?
Following Laura's point, really, at the end of the day, we're trying to homogenize our population of patients, right, for either the treatment or diagnosis. And of course, this disease is the most heterogeneous disease we're going to see. So it goes under the umbrella. So it's thought to digest, right? It's how can we homogenize these diseases when we give them a first and a last name so we can really understand what is the mechanism of action, what are the potentials up-down signals that can be used. So I'm a strong believer on studying extreme diseases. So orphan indications for the disease that you're looking for, that your drug you think is going to work. So get a population of patients that are pure, I'm an inflammatory guy, so I love inflammation. So I look for a homogeneous population of patients that have a pure inflammatory ocular surface disease, and then compare it to the non-inflammatory components, or patients or subjects that have some other form of genetic metabolic issue that has nothing to do,
or nervous system
right?
issue.
And I think, you learn from those two extremes, and then you start figuring out where you're going to hit the spot in the heterogeneous population of patients. And that takes patience too. And of course, the market size might not be that big, but certainly will lead to discoveries that will open the field for other discoveries. So
do you think if we're able to, first name, last name, define these patients better, develop better clinical diagnostics during the clinical trials development process, more sensitive, more specific, more streamlined, do you think there's an appetite with the regulatory bodies that review the data and decide whether to approve a drug or not? Do you think they're open to patient-reported outcomes, to other metrics, updated metrics, something other than Schirmer's to measure clinical effect? How much do we need to support helping regulatory bodies understand the importance of accepting patient-reported outcomes and other metrics for looking at disease activity? Because, in the UK, it's you don't have a lot of these tools.
True.
And that's like, I admire you so much. You're like Tale of Despero. You're fighting with one hand behind your back all the time. So respect to that very much. But do you think there's movement amongst the regulatory bodies in changing how they approach these things and maybe streamlining it a bit and paving the pathway to acceptance a bit more?
Yeah, not always as fast as we'd like. And of course, it's been complicated in Europe with the UK coming out of Europe and stuff like that, and new bodies looking at stuff. But again, I think that's the power of consensus of an audience like this. Actually, if industry as a whole is getting behind some of these new metrics, it's much harder for regulators to ignore that. So again, instead of working in isolation and each going for our, you see it with things like PROs, each company developing their own, that's never going to work. Gotcha. What we need to do is actually develop one together, which will help us all move forward in terms of meeting those endpoints.
Teamwork makes the dream work.
It does indeed. And again, I think it's really important that we get behind the diagnostics that are coming on board. How can we use those better in trials? And in the early days, we will probably have to do quite a few of them and see what fits with our particular population. But that will help us to narrow down in future to say these are the ones that I'm particularly targeting with my new mechanism.
So, I would say yes, for sure. The regulatory agencies want to have measures that they can make a decision that is, very easy.
Yeah, their job is safety and efficacy.
So it really is, again, go back to our first point when we started, is the investment of developing those technologies. And of course, once you validate it and show that, it shows exactly what you're looking for, and maybe when you just need to step back, we're trying to maybe develop all these diagnostic tests that are too big to analyze. And, to Laura's point, you find maybe pockets where you can actually find that signal. And regulatory agencies would love to have diagnostic testing or endpoints that can be followed appropriately, safely, and that are relevant.
So let's say as a team, as a community, with everybody leaning in together, shoveling together, that we're able to make some traction and movement in the regulatory, streamlining more tools in the toolbox. At the end of the day, as a busy surgeon and a clinician surgeon that does a lot of surgeries, right? What are the main hurdles and obstacles in implementation of some of these streamlined, directed, specific potential therapeutics?
I think as a surgeon on the diagnostic side, like MMP tests, osmolarity. I mean, the 2017 ASCRS survey showed less than 10% of ophthalmologists are utilizing those tests. So it's just a non-starter for a busy surgeon to go.
What's your guess as to why that is?
I think, well, I have my opinions, but I probably shouldn't say that. I think it's time. I think it's inter-test variability. And what do you do with the information? That's the other issue. Having said that, one of the technologies that I'm pretty excited about that has made it into our clinic are things like the eye trace, where you can measure the tear film and look at how it breaks up over time, and it kind of sends a, hey dummy, don't operate on this patient until you get their tear film in better shape.
Yeah, it almost waves a red flag in your face.
Yeah.
The eye trace, yeah, it's pretty cool. And it's fun to be able to figure out where in the optical system is the problem for,
Right.
for the patient's symptoms.
It's interesting that really only about 1 out of 10 clinical trials actually make use of the MMP9, so you're missing that one piece of, well, as Dr. Perez, he's the inflammation guy, right, Doc? But missing that one piece that will tell you a lot about inflammation on the anterior surface, and I think that is very valuable. I think we're also missing maybe some of the between study visits, and again, I'm going back because this is what I do primarily is clinical trials. We're missing a lot of that extra data that we could be giving the regulatory agencies about environmental factors, allergens that patients are exposed to, their digital screen usage. As you saw in the introductory video at the beginning of this session today, air pollution in some parts of the country may be a factor, but I believe with the digital tools that we're going to begin now using in future trials in the months and years ahead, we'll be able to deliver some real-world data that we've never had access to before, that we'll be capturing from patients with some of the handheld devices. Who knows, maybe someday even a device that can be worn by our patients to track some of these factors as well.
I was tracking MMP9 reduction with each IPL treatment session. This was about 2017. And, predictably dropping with each IPL treatment. Came in the next visit, lower, lower, lower. Well, all of a sudden, it wasn't dropping anymore, and I'm like, what's going on? I haven't changed my technique. My device is working just fine, and what's going on? And it took flying to Nashville to figure out why. This was at the end of the summer of 2017, and the entire West was on fire. As soon as I crossed the Mississippi, the air was clear. I'm like, oh, pollution, air quality index. So I went back and looked at my data again, and the R coefficient was like 0.83 with the air quality index and the elevation of MMP9, the inflammatory load. So I love where you're going and where you're thinking of like putting data, putting numbers, putting patient experience in the context of their environment, in context of the other aspects of the definition, the very definition of dry eye, including inflammation, right? And those environmental factors, the neurosensory compromise that impacts so many of our dry eye disease patients. Which leads me to something else I want to ask you guys about. AI. We talked a little bit about it earlier today, and I love the app that you've created. Is that available here as well?
I believe you can download it here.
Oh, I'm going to check it out. This is so cool. Yeah, so like when you bring on this element of AI, how much do you think it will help us really, not to be creepy, but get inside the head of the dry eye patient, of what their experience is, and use it to multiplex analyze all of these multifactorial aspects of their daily life? Can we leverage it in a way that will help us be better clinicians, better researchers?
I think, and I'm going to indirectly answer your question. I apologize in advance.
No, it's okay. I don't want to
But I think, getting back to diagnostics, I was involved in a study with Trufford Simpson and Carolyn Begley, and we looking at staining. I mean, that's always one of the frustrating things because one person's 2 on the grading is another person's 2.5, and you're trying to get a p-value of 0.05, and
With a concordance agreement
right,
of 20%.
20%. So we did a study with 5 sensors, standardized the slit lamp apparatus. We standardized things that I don't understand. I was just the clinician who was like, hey, I've got tons of dry eye patients. You want them? They're like, yes, please. And we standardized luminance, chromaticity, things that I don't understand. But it was amazing when we got these photos. We took photos 1 every 30 seconds for 5 minutes. And what we found is that there is a time period during that 5 minutes where you have enough stain, you're not quenching, you don't have too little stain, and you can actually, really dig down into how those, those particular staining, studies truly look and how they, you can get a good feel of whether or not something's effective or not. So we got a bunch of images, and then we sent it out to 10 dry eye experts, and we all took them from, everybody agreed this is 1+, this is 2+, this is 3+, this is 4+, and then we had 143 images, and the concordance amongst the 10 readers was just astounding.
How bad was it?
It was good. It was actually
it was
great.
good?
Yeah,
Okay.
like we were all, agreed that, hey, this is, worse than that one. So I think, the fact that there was so much concordance amongst the observers, I think we could apply AI because, one of the other things too is like, if there's central staining, add a half. If there's confluent staining, add a half. And these are just all huge variables. And so if we could streamline it with AI, have a centralized reading center, centralized, ways of obtaining the staining measurements, I think, that would be a win.
I've been playing around with some software by Advanced Ocular Systems that actually uses AI image enhancement technology to count the pits. And that might be another way to stratify a difference in clinical effect and placebo versus active drug in these trials as well. I would love to see more of that, and it would certainly be less expensive than having 10 different doctors come up with a good concordance rate. That is, it's hard to standardize at that level. My hat's off to you for that little body of work there.
Like I said, I was just providing the patients, and they humored me. But yeah, you think about the cost. It was very expensive to put these slit lamps and all the various and get a trained photographer. But then you think on the flip side, how many companies have tried to bring a product to market, and it was not successful? You think about the cost of that.
Yeah, so we've got to remember that things like AI can only work with the quality of data that we put in.
Junk in, junk out.
I think, not being disingenuous to our retinal colleagues, actually taking a fundus picture is fairly standard. And the OCT that you can do now is brilliant. When it comes to anterior eye, you've got different background of iris, color, textual information, etc. It is really challenging as a clinician to get good images, and then the AI's got to learn from those. But the nice thing about AI is when you do get the input well, it can tell you what's in that black box in terms of what it's particularly looking for. And we've published recently on looking at some of these grading and in fact what clinicians are doing when they're giving you that magic grade. Actually, how are they putting that together when you really look at the distribution of the staining, etc. So we are learning and we are getting better. We've still got a way to go, and AI gives us a way forward to move with that. But again, we mustn't forget about the patients. And I think one of the great things about improved diagnostics is sharing those with patients. Because when they understand, particularly when they see their meibomian glands and how they've declined with time, the motivation for them to actually use the treatment is huge. So actually having more advanced diagnostics, we all hold on to those numbers from various other systemic diseases that we have. Actually, we need the same in dry eye to help patients to become a co-partner with us in managing their disease.
This is kind of like the coming attractions part before you watch the movie, but Aura has developed a way to measure objective data in real time from our clinical trial patients, whether the patient is at home or at work. There is a way to capture this data in real time, and I think we haven't really talked about this yet, but I think as databases are created from that data, there might even be a way to do real-time grading, for example, on conjunctival redness and
Yes.
maybe corneal staining, even when the patient is not in the clinic. And I believe that's part of the future, some of the future elements of clinical trials as well that Aura is introducing here today.
I think that could be pretty simple to put together. We did a trial looking at the daily progress of chalasia on the eyelids for IPL, and patients got really good at sending in these selfie photographs. It takes a little bit of coordination in front of the mirror, but you can teach them how to do it, and I would imagine that with your phone being able to shine a bright blue light and give them a fluorescein strip and a few little steps of instruction, maybe we can capture things in between visits and capture data points outside the code center, less expensive. What do you think, Victor?
Yeah, well, AI sounds really sexy always. I think it's very important that we know exactly what we're measuring, what type of convolution or non-convolution analysis you're doing. And, again, goes back to our partnership with companies where, like you said, in Retina, everybody's been using AI for a while, right? Macular edema, corneal nerve, et cetera. So I think it's how we partner to bring that to the ocular surface. And second, I love both the idea of, patient collection of data. I think that's, the more real time. I've been involved in some clinical trials that have been very exciting where you're collecting data in real time, especially for this type of disease. This is a disease of symptoms. And we need to get better at that, how we capture that. So of course, we can put into the AI software the symptoms, but if we don't have a good questionnaire that's looking for what we're looking for, we're not gonna move forward. And we learned that from the neuropathic field. I mean, we really have not too much knowledge of exactly what we're measuring from our questionnaires and when. So that's another, hurdle that we need to continue to work. And James, you've done a great job working with questionnaires.
Yeah.
And they're not that easy because you need to validate them. But at the end of the day, it's a disease of symptoms. And that's something that we also need to focus on.
So before we do a rapid fire section, any final thoughts on what we've discussed so far? Something you want to get off your chest? No? Okay. So I'm going to ask each of you to quickly answer as quick as you can. What do you wish you had in your tool bag, your clinical treatment tool bag now in your dry eye clinic? What's the one thing you wish you had? Dr. Wolffsohn.
I think the most pressing thing for me is a way to better sample and analyze our tears in the clinic. Because again, there's a wealth of information there in theory, because the tear film, the blood vessels leak into it. It should be as diagnostic as blood, but we just don't have those measures at the moment. So that would be my top priority amongst many.
I would say something that works quickly and it works for just about everybody. I do, it's pipe dream. But having said that, look at glaucoma. I mean, there's textbooks written on glaucoma. There's how many different types of glaucoma, but what's the one common thing? Get the pressure low enough and glaucoma doesn't progress. You think about cancer treatment, before immunotherapy, it was just, cross-link the DNA and hope for the best. And, that's been revolutionary. You think about retina, you stop the growth of blood vessels and you fix macular degeneration, or at least stop it. So, I agree we need to, subcategorize each of the types of dry eye, but where is that blockbuster step that's going to give us that product?
I mean, maybe not in my clinic, but in my practice, I want to know more about natural history of disease. I mean, we really have very little data on that. It's not easy. Many of you may have some of that. So I think understanding the progression of these first and last name conditions will help a lot and will help me stage my patients. And in that same scenario, decide therapies and diagnostic tools that I may need.
I love it. Maybe even a set of prognostic indicators of how quickly they are to progress that we could develop as well. Dr. Downie?
For me, it's about a way to navigate the drug landscape better. So how do we have a tool that allows us to rapidly and accurately stratify our patients and know which drug is going to work? So really about removing trial and error prescribing as quickly and as efficiently as we can.
Take a lesson from oncology, right? Look at how customized they're becoming.
As Dr. Springs just said, just for me, it's more specialized medications, quicker onset of action, and something that will just allow us to really enhance the compliance for our patients.
Love it. And then final rapid fire, how many years are we away from a cure for dry eye?
I don't think we'll ever have a total cure.
I agree. You?
I agree. I think we are far from it. This, as Victor was saying, we are clearly in the infancy of this.
I mean, we might be far, but in the short term, I think we can do better.
There you go.
And that's, I think, the big picture. Cancer, I mean, think about it. Cancer way back needed the government to put an initiative to cure cancer and created a National Cancer Institute. So I think, all here surface and ophthalmology, we're in that track. So I think about short terms, how we do it next.
Love it. Dr. Downie?
I think, for this audience, everyone knows the importance of dry eye, but in other spheres, it's often trivialised. And that's a problem because it needs investment. And I think part of the challenge is that we've termed a really umbrella. We've got too much of an umbrella term. So we'll probably have cures, I reckon, for subtypes of the condition. And then it's going to take much longer, though, to actually get effective drugs that cover the full spectrum. So maybe a shorter term capacity for more nuanced drug therapy, and that will evolve over time as we better understand the complexity of the disease.
Well, there are a lot of those great medications, I believe, already in development now. You're going to hear about that in a few minutes, from what I understand. But I believe that we're using some of them already in Memphis and some of the trials and companies that are represented here today. So hopefully sooner than later, maybe one of those already exists. And so maybe eventually we'll all be saying together, thank you very much, as Elvis says in Memphis.
Lean in. Thank you, everyone.